right, okay.
Perfecto, muy bien. Muchas gracias, Diego. Bueno, muy buenas tardes en Buenos Aires y muy buenos días en Australia. Estamos encantados de contar en esta ocasión con la profesora Diane Nicol de Australia, quien va a dar su conferencia de ingreso como miembro de número a la Red Internacional de Bioderecho. La profesora Nicol es una profesora distinguida de Derecho de la Universidad de Tasmania en Australia. Tiene estudios en ciencia, así como también en Derecho, y cuenta con un doctorado en Biología Celular. Ella es miembro de la Academia Australiana de Derecho y de la Academia Australiana de Salud y de Ciencias Médicas. Es además la directora del Centro para el Derecho y la Genética en la Universidad de Tasmania. Digamos, tengo que decir que es una figura absolutamente importante del bioderecho mundial, y su entendimiento de las nuevas tecnologías es verdaderamente notable. Ella nos va a acompañar con una conferencia en inglés, ¿cierto? Después las preguntas que ustedes quieran hacer, eventualmente si tenemos tiempo, yo se las haré en inglés también, y traduciremos después su respuesta al español. Ella va a usar PowerPoint para que puedan ustedes seguir un poco mejor la conferencia, pero en función del tiempo que tenemos, que son conferencias tan reducidas, no tenemos tiempo de ir haciendo traducciones simultáneas como la hemos hecho en la mañana, así que esperaremos que termine, y si hay preguntas, esas sí las traduciremos. Su conferencia será edición del genoma y vulnerabilidades de las futuras generaciones. Okay, Professor Nicol is a distinguished professor of law at the University of Tasmania in Australia. Diane has a background in science as well as law, with a PhD in cell biology. She's a fellow with the Australian Academy of Law and the Australian Academy of Health and Medical Sciences. Diane is also director of the Centre for Law and Genetics at the University of Tasmania. Her current research focuses primarily on two areas: the regulation and governance of personalized medicine, genomic data sharing, biobanking, genome editing, and other emerging technologies, and commercialization of biotechnology and patenting of biotechnological inventions. I have to say that Diane is an absolutely outstanding figure of international biolaw, and her understanding of new technologies is remarkable. But at the same time, I think that is very challenging. As she has said, and I quote, "The question of how to regulate emerging biological technologies, particularly genetic technologies, is an incredible complex one. There is no clear right or wrong." I close the quote. So I think that seems to leave room enough for hermeneutics when analyzing biogenetic settings. So her thought is not only notable, but also very challenging to us. So Professor Nicol is going to give her talk, Genome Editing and the Vulnerabilities of Future Generations. Diane, you are completely welcome. We are thrilled, really thrilled to have you here. It's an honor to us. So you can start when you want.
Thank you so much, Eric, for that incredibly kind introduction. And I'm absolutely delighted and honoured to be invited to talk to you all. And I thank you, Eric, and Laura and Marissa as well for letting me have the forum here. I first have to apologise that I only have the English language to speak in, and I know that that is always the sin of the English language speakers. It's rare that we have other languages to speak with, so please forgive me for that. I'm just going to share my screen because I do have my PowerPoints, as Eric said.
Yes.
And hopefully that comes up okay for you all. When Eric asked me to talk about a focus on vulnerabilities, I thought that this really fitted in nicely with some of the work that I'm doing on the regulation of genome editing, which I'll explain in a minute, because it's so much a focus when we're looking at future generations. So the vulnerabilities of future generations is what I really want to focus on, and the legal issues associated with this. So my presentation, I'll first give a little bit of an introduction to some of the scientific and ethical issues, but then I want to focus primarily on the law relating to heritable forms of human genome editing, some of the scientific debate that's really leading us in this area, and then the law specifically relating to research with human embryos, community consultation work that I've been involved in, and final thoughts. And I'll try and do all of this in 20 minutes. So first of all, I'm sure many of you are familiar with the technology of genome editing, but I'm doing a very brief overview here. In a book chapter for a handbook that Eric is editing, my colleague Chris Rudge and I describe it as an exquisitely precise and targeted way of making changes to DNA, far more than was available in the past. There are various forms with very strange names like talons and zinc fingers and CRISPR, and we don't need to talk more about that, but CRISPR is the technology that is really leading the way. It introduces this complex editing machinery into cells so they can cut DNA and allow it to repair either naturally or in a guided way. And by these means, genes can be shut down, genes can be activated, genes can change function. And it has applications in a whole array of areas, whether plants, animals, bacteria, or indeed in humans. It's seen as having unlimited potential. But of course, different forms in humans have different ethical and legal implications, whether we're talking about non-heritable sort of forms of genome editing that are done on adults or even children. And some of the clinical trials that are going on in this area include clinical trials relating to sickle cell disease and some of the most intractable diseases that people suffer from. But there's also debate around the idea that these changes could be introduced in a way that could be heritable, passed on to future generations. And of course, research, particularly using human embryos. So these raise a whole array of different ethical issues. The main debate for non-heritable forms is to do with justice. How do we make this accessible in a way that's equitable to the community as a whole? And I'm not going to talk about the ethics generally. I'm sure there are many other people here who could talk more profoundly about ethical issues. But some of the things that are coming up are things like when we're passing on things on to future generations, so little is known about the risks of physical and psychological harm. There's a lot of debate about the slippery slope to designer babies. And of course, the moral status of the human embryo, which features very much in our Australian debates and probably also in many other countries, but not necessarily all. So looking at the legal issues then, it's interesting that there's very little in international law to guide us. I think the most prominent example is the Oviedo Convention, which talks specifically in Article 13 about intervention seeking to modify the human genome may only be undertaken for preventative, diagnostic, or therapeutic purposes, not for enhancement purposes, and only if it does not aim to introduce modifications in the genome of descendants. So that's a clear obligation, certainly in European law. I just want to refer to some of the analysis of domestic regulation that's going on. And I'm referring specifically to a book, an edited book that I was involved in, edited by Andrea Boggio, and Romeo and Almquist. So Boggio, Romeo and Almquist got together a group of authors to look at an analysis of how human genome editing is regulated both in heritable forms, but also in relation to human embryos. And you can see there's a whole mix of types of regulation. And I'm going to focus specifically on my own jurisdiction, Australia, but Australia sits alongside many other jurisdictions where there's a clear legal prohibition. There's de facto prohibition by regulation in some countries. Some are much more open, and even in those that prohibit, there is some leaving the door ajar, leaving the opportunities available in the future. And for others, well, this book does not include most of South America, for example, but I'm sure you could give me insights as to how things are regulated there. So in Australia, there is a very cautious, very strict prohibitory regime here. This came about in 2002 when we enacted the Prohibition of Human Cloning for Reproduction Act. This was in response to advances like Dolly the sheep, for example, the concern about human reproductive cloning, but it covers a whole array of other issues as well. For example, Section 15 makes it a criminal offence to make heritable changes to the human genome with 15 years imprisonment. So this clearly prohibits HHGE, as I've used it for the acronym to mean heritable human genome editing. But we actually have a bill that is in our parliament at the moment that is seeking to allow mitochondrial donation, which would allow changes to the mitochondrial component of the human genome if it's approved. So those are the sorts of examples of where the law is. But I want to turn now to the views of science experts. So even though CRISPR technology is less than 10 years old, I think this recognition from the scientists that they have unleashed this capacity for incredible developments, but also the capacity to unleash the prospect of doing things that might be considered to be unethical. And so science experts have really got together globally to make statements on the way they see they should be self-regulating their own science. When it comes to basic lab research, they see that we should use existing regulation. For non-heritable forms, use existing regulation, but it should be limited to disease and disability. And clinical trials are important to evaluate safety and efficacy. That probably goes without saying. It's using existing forms of regulation. When it comes to heritable forms, they're certainly not closing the door on this, but recognize it should be only permitted for compelling reasons. For example, only where this is the only way to possibly have a genetically related healthy child, but there should be strict oversight, and it should only follow public discussion, which relates to the last point I want to get to today about community consultation. And as for enhancement, we shouldn't proceed at this time, but there should be public discussion and policy debate. Then in 2018, I'm sure many of you will know at an international congress in Hong Kong, this scientist, Professor He Jianku, announced that he had actually used CRISPR technology to modify a gene relating to immunity against HIV, the so-called CCR5 gene, had done this in a number of human embryos, and he claimed that this had resulted in at least 2 live births, twins, and later it was adjusted to say there were 3 live births. As of 2020, it was announced that he had actually been imprisoned for 3 years for illegal medical practices. And that's as much as we know. We don't know what the consequences were for the live births. But of course, this caused a firestorm of criticism and debate and new calls from the scientists that there should be a global moratorium. But the organizing committee was still concerned about a blanket ban on heritable forms of genome editing, or germline genome editing, as it was called. They said it could still become acceptable in future if risks are addressed and if a number of criteria are met, including attention to societal effects. After that, an international commission was created to look at clinical uses of heritable human genome editing. They released their report in 2020, and again, a very cautious approach, leaving the door open, but recognizing this would be in some time in the future if it was not otherwise possible to create genetically related offspring free from serious disease. So only in a limited context with a set of strict criteria and assurances of safety and benefit, and again, after societal debate. The World Health Organization also has a global group of experts that have been looking at the issues relating to human genome editing generally. I've not put up their guidance, which was released earlier this year, because they don't talk specifically about HHGE, but it's also important to take into account what they're saying. So from the scientists themselves, we're getting this idea that there should be a very cautious approach, and that there is a need for more research and a need for societal debate. So what then of research? What research can be done? And I put here the ways that research can be done, particularly if we want to look at heritable forms of genome editing into the future. And of course, there are a whole array of types of research that can be done, starting with animal models and human cells in vitro, moving to human clinical trials of non-heritable forms of human genome editing. So all of these things are going on at the moment in a regulated fashion in most jurisdictions. But one of the really problematic sticking points is that if we ever do want to move forward on heritable forms of human genome editing, it's really going to be important to be able to undertake research involving human embryos. And certainly in my country, that raises the really significant community concerns and ethical concerns. And there's an array of types of human embryos that could be examined for this purpose. First of all, embryos unsuitable for implantation. Also, embryos that are excess to the needs of families for artificial reproductive technology purposes. Embryos not created by fertilization. There is a great deal of advancement in the development of blastoids, embryos or embryo-like entities that are not created by fertilization. And of course, the final point is embryos created by fertilization for research. There is justification for why this is needed, but of course it raises really significant issues. I go back to the Boggio and et al. Book, and they also looked at embryos created by fertilization for research. Of the analysis, in countries like my own country, creation of embryos by fertilization for research is prohibited. It's permitted in other countries, and then there's a whole array of other countries that we don't know so much about. The Australian position, Section 12 of this Act, makes it an offence to create a human embryo by fertilization other than to achieve a pregnancy of a woman. So clearly prohibits creation of human embryos by fertilization for research. But this mitochondrial donation bill might, if it goes through, would allow this to happen. There is another piece of legislation in Australia, the Research Involving Human Embryos Act, which does allow creation of, does allow certain forms of embryo research, but only under license. from a body called the Embryo Research Licensing Committee. And this lists the types of research that can be undertaken. If they're not undertaken under license, then there are criminal offences associated with this. I won't go through each of them. I'll obviously leave you with the slides, which gives an indication of these things. So the apparent consequences for countries like Australia then are that we can't undertake genome editing if it makes heritable changes. Human embryos can only be created by fertilization for ART, and we can only use excess ART embryos for research if under a license. The problem from the perspective of doing genome editing research is that the excess ART embryos are often frozen embryos which have undergone a number of cell division cycles. So they're probably too advanced for useful human genome editing research. We could also possibly use under license embryos unsuitable for implantation, pre-embryos, that's zygotes, for example, up to the first mitotic division, and embryos created by processes other than fertilization. So there are things that can be done in countries like Australia, but it's certainly not the case that you could create embryos for research by fertilization at the present time. So if we look at the law then on heritable human genome editing, it's prohibited in some countries and research is limited. Others rely on more soft laws, others are more open, as we've seen, and others we know less globally about what the laws actually say, if indeed there are laws. International science policy is leaning towards future approval, but strict limitations and conditions. We need more research, and it's clear from the scientific community that they recognize community views are important. And I just want to talk for a couple of minutes about some of the work we've been doing in Australia on community views. If we do need law reform, it's really important to bring the community into the conversation. And I've been working with a group of scholars on citizen deliberation, looking at how we might engage with members of the community and how we can understand what community views are. The challenge is, of course, this is really complex technology, and it's not the sort of thing that you can engage fully with the community by surveys or other forms of sort of normal community dialogue. So what we've been proposing is more to use citizen deliberations. And we've been proposing an Australian citizen jury on genome editing and a much broader and more ambitious global citizens assembly to ask the question, under what conditions or circumstances might the application of human genome editing technology be acceptable? And we've been looking at both heritable and non-heritable forms. I don't think I've got time to go through all of this, but the goals of our Australian citizens jury was really to create a forum where ordinary citizens, through deliberation, can develop their understanding of human genome editing, explore their positions, and formulate shared recommendations on ethical, legal, and social aspects. So this was the really dominant theme of the work we've been doing. And these are the various phases of the work. I conducted 34 expert interviews last year, and from that we did discourse mapping. Selected participants did interviews, and we held our citizens jury in July this year in Canberra. We were really, really fortunate in that we were between pandemic lockdowns, so we were able to bring 23 members of the Australian public together to debate these issues. Then post-jury interviews followed, and we're at the research phase now, and I just want to talk a little bit about that. Participants were selected for demographic and discursive representation. So it's demographic, but we also wanted to select people based on differences in views about the technology. And my colleagues have this methodology for looking and selecting based on discursive representation. So as a result of the citizens' jury, we're doing our preliminary analysis at the moment, but we do have a set of broadly shared positions on human genome editing generally that we believe our group of participants generally agreed to. And their focus, as not surprising, as the idea that it should be to alleviate human suffering, improve quality of life, and reduce childhood mortality. This was really important to all members. Recognize that adequate research is needed, and for them, the overarching ethical principles should be meaningful consent and equity of access. They recognize that research and clinical applications should be subject to appropriate regulation and approval processes, which involve community input. And they said, look, this is so complicated. We need key terms to be defined so there's clarity. This reduces the possibility of miscommunication and misuse. And recommendations should be revisited as the technology advances, and the technology in this area is advancing so rapidly. Views on heritable forms of editing, and I say that this is the majority of their view. For some people, there was a deep-seated view that they couldn't ever contemplate allowing heritable forms of human genome editing. The majority had a more supportive stance for future applications, but they all agreed that it should be very limited to where it might assist people to have healthy, genetically related children. But there were diverse opinions as to what constitutes appropriate regulation. And the embryo research preference for alternatives, but they felt that the door should possibly be open to creation of human embryos through fertilization, but subject to stringent regulation and oversight. And they maintained that the 14-day rule, the 14-day limitation should remain, even though there's an international view that it should be relaxed. Next steps are other national juries and global assembly. But my final thoughts on all of this is that the law relating to heritable human genome editing, as we've seen in some jurisdictions, is very prohibitory, both of heritable forms of editing and research involving human embryos. But this is not the case everywhere. International reports recognize the need to think deeply about this, including societal debate. The indications from the majority of our citizens in our citizens' jury is this support for property regulated, equitable and fair, non-heritable forms of human genome editing, possibly even heritable, but conditional on more research and consultation. There's also some tentative and cautious support for properly regulated creation of human embryos for research. And we really await deliberations in other countries because this is an area where the global dimension is so vital. Finally, I just want to acknowledge the funding agencies. Our funding is from Australian Research Council and the Australian Medical Research Futures Fund. I should also note that I am a past chair of our embryo research licensing committee in Australia, but I speak entirely in my capacity as an independent law academic. And I just want to gratefully acknowledge the contributions of both of the groups that I work with. And I apologize if I've gone a little bit over time, Eric, but I hope I've left a little time for questions.
Thank you very much, Diane. The timing was perfect. Thanks a lot for your very excellent presentation. We really appreciate it. So I think we have time enough only for one question, so regarding your presentation. So that there many positions regarding genome editing, CRISPR-Cas9, so but new biotechnologies. There are some very liberal, other ones are very conservative. So there's one position that is very relevant to me, is that by Julian Savulescu. You know, he's
I know Julian's view as well.
He's Australian. You know,
yeah.
so
Yes.
and his position is very liberal. You know, so he even agrees with dysgenic practices eventually. You know, so but by considering all positions possible regarding these matters, what is your own position? For example, which uses should we give to genome editing? Do you agree with eugenics or even dysgenic practices?
I don't often like to give my opinion on these things. Look, I actually find it really hard to contemplate even the idea of engaging with these sorts of practices. And to me, from using my scientific hat, from the scientific perspective, we are just so far away from even contemplating those sorts of things, you know, contemplating the idea of being able to do them safely. So to me, it's not even a debate that we should be getting into. The debate that we should be having now is how we actually achieve the things that are achievable at the moment safely and ethically. So, for example, when we're looking at non-heritable forms of human genome editing, how do we make sure we follow basic principles of beneficence and non-maleficence and justice, particularly in dealing with those issues? So that is my position, that we need to have much more debate about how we implement what is achievable now before we get on to these really speculative ideas about eugenics and the like. Even the idea of safely practicing heritable forms of human genome editing to alleviate human suffering that many families face, I still think that that is such a long way off before we can do that safely. So these are interesting theoretical debates, but to me, they're entirely theoretical at the moment.
But in some way, it's also practical, because CRISPR-Cas9 is a very revolutionary practice. In fact, Doudna and Charpentier got the Nobel Prize for that. And so it's not only theoretical discussion. I think it's very practical as well, at least in some way. So, bueno, le pregunté a Diane, considerando todas las posiciones que existen liberales y conservadoras con respecto al CRISPR-Cas9, o sea, la edición genética de seres humanos o de cualquier organismo, ¿cuál es su posición respecto a esta? Porque hay posiciones muy liberales, por ejemplo, que incluso aceptan el uso de estas prácticas con fines eugenésicos o digenésicos, incluso para desmejorar ciertos individuos. Ella dice que a ella no le gusta mucho dar su opinión personal con respecto a estos asuntos, pero que también le cuesta contemplar realmente que estas cosas puedan ocurrir sin antes, digamos, una discusión o mayor debate respecto a estos asuntos. Que su posición de un punto de vista objetivo sería que deberían entrar en juego varios principios de la bioética. Ella menciona beneficencia, no maleficencia, justicia en este debate, y que claramente tendría que haber un debate muy profundo con respecto a una eventual implementación de estas prácticas, incluso con fines terapéuticos. Diane, thank you very much for having participated with us. So right now you are officially a member of the International Network of Biolaw, and we are really proud of that. We are really proud of you. We are completely sure you will be a great addition to our work and to our network, and we're really grateful of you. Thank you very much for having participated with us.
Thank you very much, and I'm really honoured to be a member of the network, and I really will enjoy participating in it in the future. Hopefully, one day face to face.
Yeah, we hope the same. We hope and trust the same. Thank you very much, and take care of yourself, and see you soon. Thanks a lot.
Yeah. Thank you. Bye, everyone.
Bye. Listo, estamos listos entonces.